Click the bell icon and subscribe to our channel so that you will not miss any classes.
Hello, welcome to class. From this class, we shall discuss a new unit from second year, that is biotechnology. Unit 9, biotechnology. And in this unit, biotechnological topics will be studied under two chapters, that is chapter 11, biotechnology principles and processes. Principles and processes, and chapter 12, biotechnology and its applications. Here, the principles involved and the processes involved in biotechnology techniques will be studied in the first chapter, principles and processes of biotechnology, and from those biotechnological process, what application we are having, what usage we are having current day. It may be in daily life, it may be in agriculture, it may be in medicine, it may be in any other fields. Whatever the applications we are having, that will be studied under the chapter biotechnology and its applications. Now we shall take up the first chapter, that is biotechnology principles and processes. Before starting it, the biotechnology term was coined by Carl Henry. And biotechnology, to define it, what is meant by biotechnology? To understand it clearly, we have to understand some basic things, or we need to discuss some basic things what we know already. That is, I'll give some examples or some processes so that you will understand what is meant by biotechnology process. So here, the making of curd or idli, dosa batter, and making of wine and rearing of livestock, that is rearing of buffalo, cow, sheep, goat, hen, for getting the meat or the egg. So these process were age old. That means once the evolution has started happening in human life, that is, when we see the evolutionary growth of human beings, at various stages, human have started various process for his betterment. For example, in order to improve food, they started making curd, they started making batter, and they started making wine, and they also started making rearing the livestock. And also, there are many improvements seen in these. That is, that is making of curd, making of batter and wine are the microbial mediated processes. And there are various techniques developed today in order to improve the product quality also. Which means they are using some technique in order to get something for our betterment. So this is also a sort of biotechnology. which means biotechnology is not today's science. The term biotechnology is very recent, but the concepts of biotechnology are age old, but it is developing today. That is, it is getting advanced today with the advancement of biotechnology. This is one shade of biotechnology. But nowadays, biotechnology term is used in a restricted way. That is, creation or alteration of gene. Creation or alteration of genes to get products and services. So once we see or once we heard the term biotechnology, what comes to in our mind or what is developed is, it is just a creation or the alteration of genes in order to get the products and services. Not only this, there are various advanced techniques available in biology, that is in vitro fertilization, to get a test tube baby, and development of vaccines. So these are all the kinds of biotechnology, but there are two shades. It is traditional way and it is modern way. This modern way is because of development of advanced techniques. So here, before also agriculture was there, but the techniques are not much advanced. But here the techniques are much advanced, so we are referring it as modern biotechnology. So we can give two shades for biotechnology. That is, it could be modern biotechnology or it could be traditional biotechnology. So this is traditional biotechnology and this is modern biotechnology. And now here in this chapter we are going to discuss what are all the advanced techniques developed in modern biotechnology. That is, how the genes can be altered, how it is possible to create a new gene, or how it is possible to develop the vaccines. Those process we will be studying it in detail under the chapter biotechnology principles and processes. Now you may ask the question, here in traditional biotechnology also there may be a chance of gene alteration, the things like that, of course, but before, that is in a 20th century, before 20th century, the structure of DNA was not known. That structure of DNA was not explained and the composition of DNA was also not clear. So hence there was no much advancement in this process. But in 20th century onwards, that is mid of the 20th century onwards, there was a drastic development in the biotechnological processes because there was a discovery of structure of DNA, with the structure of DNA well explained in 1953. That is in the middle of 20th century, there was a drastic acceleration given to the modern biotechnology. Hence, mid of 20th century onwards, it is called golden era. It was called golden era in biology because of the discovery of structure of DNA. With the discovery of structure of DNA, it was possible to study the characteristics of genes, composition of genes, how the gene can be altered and how the gene can be transferred from one organism to another organism in order to get a useful products. So here we are going to discuss those things in detail here onwards. We have discussed there are two phases in biotechnology, that is one traditional biotechnology and modern biotechnology, but the definition for biotechnology is given by including both of these, and it was given by EFB. EFB refers to European Federation of Biotechnology. European Federation of Biotechnology has given the definition for biotechnology by including both natural or the traditional biotechnology and the modern biotechnology, as it is the integration of natural science, cells, parts thereof, molecular analogues, to get products and services. That is, I told European Federation of Biotechnology has included both traditional and modern biotechnology to give a proper definition. That is, it is an integration of natural science. Without a natural science, nothing is possible. No doubt we are talking about DNA, structure of RNA, the things like that, the advanced one. But all of these are existing in a natural science. So, it is an integration of natural science, cells, parts of other organisms. It may be a leaf of a plant, flower of a plant, seed of a plant, anything, or any organ of an organism. It may be. And the molecular analogues. Molecular analogues refers to DNA, RNA and proteins. They are called molecular analogues. If we use any of these in order to get products and services, then it is called biotechnology. That means we are using a modern technique in order to get the products and services from these. It could be natural science, it could be cells of an organism, or it could be a part of organism, or it could be the molecular analogues. From these, we will use some techniques and we will get products and the services. And depending upon which application we are having, the biotechnology is of two types. One is product biotechnology and service biotechnology. Product biotechnology and service biotechnology. That is, if we are getting any product out of these, that means it could be cells, what I told you. So from these, then we will call it as product biotechnology. That is, for example, if we get a hormones like insulin. For insulin production, they make use of a bacteria, which is a cell. From that cell, using a modern technique, we will get insulin. And insulin is the origin of human beings. So we are getting a product. So it is called, production of insulin is called product biotechnology. And production of some other hormones. and production of vaccines. So these are all called the product biotechnology. And service biotechnology. If any of these are used in order to get a services for human being. For example, we are facing many problems, and one of the problem the world is facing is the production of sewage. That is the domestic liquid waste whatever produced upon a daily usage is called sewage. And it is having a many organic waste, and there are many microbial content which may be pathogenic, and if it is released as such, then there may be a pollution of different types of the matters in environment. Water pollution, soil pollution, air pollution, anything may happen. So one of the problem associated is the treatment of sewage. That is how to treat it, how to clarify it. For that, the biotechnological process has given a solution. So the sewage treatment upon biotechnological process, if we are treating a sewage, and we will be discussing in the upcoming chapters how the sewage treatment is done. So the things will be studied there. So we are getting a service out of biotechnological process. So we are calling it as a service biotechnology. And clearing oil spills. Oil spills. So on ocean, during the transport of the petroleum products, that is during the import of petroleum products, sometimes there may be a spill happened over there, and if this oil spills remains there for many days, then definitely the flora and fauna present in that ocean will be affected. So it should be treated as soon as possible. So it is not an easy task. For clearing that, there was a creation of superbug. That is, there was a creation of a bacteria by biotechnological process which can treat the oil spill over the ocean. So that is also a service. So there are two phases. It is product biotechnology and service biotechnology, and we will be discussing those, what are the products and what are the services we are having, still more detail in the applications of biotechnology. For as for now, just there are two types of biotechnology based on the application what we are having. Product if we get, then product biotechnology. Service if we get, then it is called service biotechnology. We have understood that in biotechnology we are altering the genes, or we are creating the genes, or we are transferring the genes from one organism into another organism. While doing so, there are many processes and the principles involved. Whenever we are conducting any experiment, there is a principle involved. So here also, in each and every biotechnological process, there is an involvement of some principle in it. So we shall discuss now what are the principles involved in biotechnology. That is, the principles of biotechnology. No doubt, there are many principles involved in biotechnology, but the two important principles or the two core processes of biotechnology could be, that is, it will be common for any type of biotechnological process or any type of biotechnological experiment. There are two common or two core processes involved, and those two core processes of biotechnology are one is rDNA technology biotechnology, and or it is also called genetic engineering. I hope you may be familiar with this, that is genetic engineering. That is, at the genetic level, the alteration or the creativity will be done. So that is called genetic engineering, or it is commonly known as recombinant DNA technology. And the second one is creation of sterile, we can call it as maintenance, not creation, maintenance of sterile means contamination free ambience for the growth of recombinant cell to get the desired product. That means in recombinant DNA, what we do is we may construct a DNA which is having a foreign gene. For example, I am telling here insulin gene from human beings. That is, from human beings, we will isolate the insulin gene, and this insulin gene will be introduced into Escherichia coli bacteria. Remember, it is from humans. From humans, we isolate the DNA which is coding for insulin hormone, and this DNA will be introduced into Escherichia coli bacteria. So this is called recombinant DNA technology. Okay, and the next step will be we have to get the insulin protein from Escherichia coli. For that, we need to grow it in a large container. This is container. So in container, we have to grow this Escherichia coli in order to get a product. What is the product here? Insulin. Imagine, except Escherichia coli, if any other organisms grow, when we transfer the Escherichia coli into the container for growth, imagine what happens if other organisms grow along with it. Along with insulin, any other type of organisms or other proteins may also grow there. Any toxins may produce over there. Is it be a good thing? No, not at all. So, that I told, there are two core processes. One is creation of it, the recombinant cell. After constructing a recombinant cell, in order to grow that in a sterile condition, we should not allow any other organisms to grow along with it. So if it happens, then our work here will be not of use. So how much this process is important, this is also that much important. That is creation of a sterile environment in order to grow the recombinant cell to get a desired product. So these are the two core processes where biotechnology scientists are working. So major working is done on how to construct a cell, and that much importance is given to how to create the sterile environment in order to get the desired product. From the next class, we shall discuss in detail what are the process in or what are the tools used in recombinant DNA technology, and how the concept of recombinant DNA technology was develop.